Thank you very much, Alain and Hugues, for the invitation, and also, and mostly for organizing this wonderful meeting. It was well worth the wait. You asked me to talk about CAR T cells, and I will, and I'll give a very CAR molecule and CAR T cell centric view of the field. Here are my disclosures. I'll be very brief on history, because we've already heard a lot about CAR T cells at this meeting. Suffice it to say that this field was born on the fringes or the outskirts of immunology, and of course draws a lot on what we learn about immunobiology and the tumor microenvironment. But really it's predicated on two things. One is genetic engineering for the purpose of instructing T cells, and secondly on the use of not the physiological receptor for antigen, but rather a synthetic receptor that some years ago we termed CAR. CARs do not recognize HLA peptide complexes. They engage structures on the surfaces of cells independently of HLA, which makes them of interest for immuno-edited cancers in particular. They can engage protein, glycoproteins, carbohydrates, or glycolipids. For those who were at the beginning of this field, there was the necessity to not only study those in vitro and in mouse models, but to engage in cell manufacturing, which really is not just manufacturing, it's really a science. We still have so much more to learn on how to grow and expand properly T cells. And to convince the world that this strange form of immunotherapy had any relevance, in fact, it had to go to the clinic, and it had to, therefore, all of this work had to fit in a regulatory mold such that the first clinical studies could take place. These are the first two CARs that were approved by the US FDA in 2017. They are both dual signaling entities here, as you see in the cytoplasmic domain, but both of them are specific for CD19, a cell surface molecule that we had identified at MSK back in the mid-90s as, at the time, just a candidate for our developing these molecules in the event that someday we went to the clinic. We thought that lymphomas and leukemias would be a good place to start. And we favored it over CD20, which was already a famous molecule back then because of the rituximab antibody, and favored it in particular because simply because it's expressed at a higher level. And this turns out to have been, I think, a good intuition, and I'll come back to this topic later. They are dual signaling CARs. These are the two prototypes, if you like. The one first described from Sloan Kettering makes use of the CD28 cytoplasmic domain, which we had introduced when we realized that the zeta chain alone was not sufficient to drive a proliferative response in T cells expressing only a zeta chain fusion receptor. Another variant that we and others had, but first reported by Dario Campana, made use of the signaling domain of 4-1BB, a member of the TNF receptor family, a costimulatory receptor that in particular plays a role in memory and also in metabolism in a different way than CD28. Following these early reports, we provided the first demonstration that you could take human peripheral blood T cells, engineer them with a gamma retroviral vector encoding a cDNA coding for this synthetic receptor and cure mice that harbored various lymphomas and leukemias. We and two other groups went to the clinic. The group at the NCI was the first to report on a case report of a patient with NHL, non-Hodgkin lymphoma, who had received an infusion of these T cells together with IL-2 and had gone into a complete remission. The group at University of Pennsylvania was the first to report on 3 patients with chronic lymphocytic leukemia, CLL, 2 of which also showed a dramatic response to a single infusion of CAR T cells, and we were the first to report on this approach in acute lymphoblastic leukemia, again in patients with refractory disease. And here's an example of what we reported way back then, seeing here the CD19 positive, mostly blasts in these 2 subjects, and you see the complete disappearance of any CD19 positive cell within days following the infusion. And I think, as you all know, these results held up in larger trials and also in the hands of other centers, quickly convincing the world that this was not just a chance observation. There are today 6 CAR T-cell products that are approved by FDA. 4 of the 6 are specific for CD19 and 2 of them for B-cell maturation antigen, a target of high interest in multiple myeloma. 4 of them use this 4-1BB structure that I mentioned, and 2 of them the CD28. We reviewed this last year for the anniversary of the European Journal of Immunology. There were at the time 700 CAR trials listed on clinicaltrials.gov. There are 900 today, but at the time about 40% of those targeted CD19, a percentage that should diminish over time as new targets enter the clinic. But we're not here to sing the praise of this approach, but rather reflect on the toxicities and the failures. The toxicity that we had, of course, envisaged, was B-cell aplasia, since a CAR T-cell could not distinguish CD19 on lymphoma or normal B-cells. And of course, it does occur in patients. We had reason that it could be manageable, and indeed it is. And in most patients, anyway, B lymphopoiesis recovers at some point. But these 2 toxicities, which are today's main toxicities, had not been anticipated from the mouse models. One known as the cytokine release syndrome, and the other one, neurotoxicity, now referred to as ICANS. As you can see, these are now codified with grades and standard procedures to manage these complications. What I'll do today is try to touch on 4 aspects of what we believe could further improve these CAR therapies. One is dealing with these toxicities. The second is improving the sensitivity of CAR T-cell recognition of tumor antigen. The third is augmenting the functional persistence of engineered T-cells. And if I have time, I'll go very quickly to mention something about novel CAR T-cell sources. We know quite a bit of about the pathophysiology of the CRS. It had been thought at first that the cytokines that mediate this syndrome came from T-cells, but owing to really mouse models that ultimately were established, it was realized that many of these cytopathic cytokines, IL-6 especially, come not from the CAR T-cells, but from myeloid cells. In these models, we also realized that IL-1 was also a byproduct, if you like, of CRS, and I'll in a minute tell you what we have done about that. In contrast, though, the neurotoxicity is still not very well understood. We know that there is often some damage to the blood-brain barrier. There is some activation of microglia. There are cytokines that can extravasate into the CNS. One thing we know is that in most cases there are very few CAR T-cells that can be found having crossed into the CNS. But the pathology remains still rather poorly understood. When we established this model of CRS in immunodeficient mice, we and another group that published a kind of similar model realized that IL-1 played a big role, not just IL-6. And in fact, preventive treatment of mice with anakinra, a soluble IL-1 receptor antagonist, diminished death from CRS in both of these models. Of great interest to both groups was the fact that anakinra, being a small molecule, it also crosses the blood-brain barrier and might therefore also tame, if you like, microglial activation in the CNS, unlike the antibodies to IL-6 receptor, which don't cross the blood-brain barrier. This has informed the design of a trial at Sloan Kettering, and I know others have started in the US, where we provide this IL-1 receptor antagonist from the beginning of the CAR T-cell infusion in 2 cohorts, and this I'm just very quickly mentioning data that my colleague Jay Park, who runs the trial at Sloan Kettering, has presented at ASH last year. And the short of it is in the first cohort of these patients, we already see a diminution of the frequency of severe neurotoxicities in comparison to historical controls, and We are now into the second arm of this study and hope that these early findings will hold up. Clinical trials have revealed many of the limitations and shortcomings of CAR T-cell therapy, and you could classify them in maybe these four buckets. One is insufficient functional persistence of the T-cell. In some patients, for example, with chronic lymphocytic leukemia, T-cells that are harvested are often of poor quality at the get-go, but we all realize that much of the decline, the functional decline of these engineered T-cells owes to either the culture conditions or the CAR itself. Another limitation owes to relapse associated with antigen escape, but here I want to distinguish two forms. One where there's antigen-negative disease, which of course the CAR T-cell could not target, and the other one where there's a relapse with still presence of antigen, and I'll be discussing this quite a bit today. The third is obviously linked to microenvironmental suppression, and I won't say anything about that. We've heard several wonderful talks on this topic yesterday. And the last one, which again, if I have time, I'll quickly mention, has to do with production costs and distribution and what we could perhaps imagine to address this problem. So first I'm going to discuss CAR T-cell sensitivity, and we became aware of the importance of this question in reviewing clinical data from various centers, and for example in BALL studies targeting CD22 with a CAR specific for CD22, it was noticed that a certain number of the relapses were characterized by diminished CD22 expression at the cell surface of the leukemia compared to pre-treatment, and you see that here. This has also been reported, and mostly been reported in myeloma, and here's an example, BCMA level on the pre-CAR T-cell infusion tumor, complete remission, nothing detectable, relapse, but relapse with a lower level of BCMA, and you see this again in some patients in a larger series. And thirdly, with CD19, we also see variable expression, and while typically in ALL, pediatric or adult, there's a pretty high level of CD19 comparable to normal B cells, we note that in DLBCL this is more variable and often lower, and I don't know if this has any connection to the 7 categories that Lou mentioned this morning. And you can see again these number of molecules per tumor cell pre-treatment here, most of them being above 1,000, but at the time of relapse, plenty of those being just in the hundreds. As I said a few years back, this made us realize that it was worth further investigating the sensitivity of these receptors. And first we compared CD28 and 4-1BB CARs, the two prototypes. And we found, and that's published in this paper, and I won't review that today, that the CD28 CAR is more sensitive, generally speaking, to a 4-1BB CAR. And this was done by generating variant leukemias with decreasing levels of CD19 at the cell surface. And this was confirmed by 2 other studies by Crystal McCall at Stanford and John Maris at CHOP, who also recently compared these and reached the same conclusions. So certainly not all CARs are equal, and these again recognize antigen through the same scFv, but only differ in their signaling machinery. So there's a difference between CARs. I'm sure there's a difference depending on the scFv, the epitope, and the affinity, but there certainly is a difference depending on the signaling machinery. So what can we do about this? And there are 2 stories I want to quickly walk you through today. One where we wanted to help the CAR, and the other one where we started searching for alternative structures that perhaps could afford greater sensitivity. So strategy number 1 here, which is to help the CAR. So in this case, this CAR here is specific for BCMA, and this, what we call a chimeric co-stimulatory receptor, is specific for CD38. It's not a CAR because it doesn't have a zeta chain or any ITAM. It's not an activating molecule, and hence the name of chimeric co-stimulatory receptor. There's a lot of interest for us anyway in mixing and matching different co-stimulatory molecules between these CAR and CCR pairs, which I won't walk you through today. The point I want to make is, as you see here in vitro, when exposing human T cells engineered with these CARs and CCRs, and we're very rigorous at expressing them at the same level. You see that in this tumor model with fairly low level of BCMA, the CAR alone only kills to this level, but with the CCRs, the killing is much better. And with the three combinations, you see the same. In fact, if you delete the signaling domain of this CCR and just retain the cell surface anchored SCFV, you also see this increase, which is what we expected, that this is this in vitro scenario, in the immediate act of killing, really owes to providing increased avidity of the T cell for the target. But the killing is mediated only through the CAR. The costimulatory domain does matter, and I just want to mention briefly that what it does, having the CCR in addition to this CAR, does improve the response here in a mouse model where we deliberately give low levels of CAR T cells, and you see greater accumulation and persistence of these T cells. That's all in the paper. I'm just showing you an excerpt here. And that is the function that we expected from providing a 4-1BB signal in addition to these CD28-based CARs. And here's another example unpublished that I want to show you in AML, where we are now targeting AdGRE2 as the CAR target and CLEC12A as the CCR target. And so here are these mouse models again, and here's the first cell line, MOLM13, much used in this field. It has a decent level of AdGRE2 expression. You see that on this x-axis, and the CAR or CAR plus CCR are not really different. Then we have this other variant where you see now the AdGRE2 level is less. And now you start seeing that the CAR barely controls and then fails, but the CAR plus CCR is better, and you see this effect on survival. And here's the lowest cell line, barely positive by FACS, and you can see now the CAR fails miserably, but the CAR plus CCR does a pretty good job. And so this is one approach of helping the CAR, and then this should go to a clinical trial at Sloan Kettering by September or October. Another idea was to just change the structure completely. And for that, we went back to the T cell receptor, which you recognize here on the left. So here are the two alpha and beta chains embedded in the CD3 complex. And to the right, you see the CARs, as we've been talking about for the past few minutes. And you see here the heavy and light chains from an immunoglobulin in these very beautiful Ukrainian colors, I note. And the question was, could we take advantage of the T cell receptor structure, which is notoriously sensitive for antigen, to target independently of HLA? And therefore, could we create a structure of this kind where the V alpha and V beta regions are substituted with immunoglobulin domains? And so this is HLA dependent and CD3 dependent. The CAR is independent of both. It's an autonomous structure. And here we would have potentially a receptor that's HLA independent, but still CD3 dependent. And we had good reason to think that this might work if you engineer it properly. Of course, these genes are all family members, related, members of the immunoglobulin superfamily, but also if you just overlaid them, if you saw here the alpha and beta chain and heavy and light on top of an HLA molecule, there's really a pretty nice fit, as expected from basic immunology. So how would you proceed to engineer this? A few years ago, we took advantage of CRISPR-Cas9 to ask a question that I won't discuss today, which is what difference does it make as to from which locus and which promoter the CAR is expressed in a T cell. Retroviral vectors, lentiviral vectors, DNA transposons, as you know, they all work very well, but they result in semi-random integration and variegated gene expression. But with CRISPR-Cas9, for example, we could now control the site of integration. And we found this track locus, which encodes the alpha chain of the T cell receptor, to be very well suited for this purpose. Regulating the level of CAR expression at essentially a good level and delaying, for example, the onset of exhaustion in these T cells. But here is just the method that I wanted to mention, and this time we go to the same track locus. We introduce the heavy and constant beta region fusion cDNA, co-expressed with the light chain, part of the exon here of the constant region, and in situ in each and every T cell, this fusion is recreated. And we were hoping to see this structure, and that's what happens. Here's an example from human peripheral blood T cells. The first control here stands for TCR alpha beta or CD3, and there is no CAR. As you target with your guide this track locus, you can see the cells becoming CD3 negative, as they are no longer able to assemble a T cell receptor complex. If you knock in the CAR, as I showed you in the previous slide, you now get CD3 negative cells that express the CAR. And if you introduce this structure, you now see these cells becoming, again, cell surface positive for the CD3 complex, and you detect these light chains at the surface. So there were many questions behind this, but the one of interest today is the sensitivity. So we created panels of clones, many variants of the NALM6 acute lymphoblastic cell line with decreasing levels of CD19. And here's from, each dot is from one experiment at one ratio of 10:1 in a 4-hour cytotoxicity assay, and you see when the CD19 level is high, both the track hit and the track CAR kill well. But as this level decreases, the track CARs no longer kill, but the hits do better, and here's the data on the right. We've quantified these, the number of molecules on these cell lines. The wild type has about 25,000 molecules, but they go down to 2,220 or so. Below 1,000, really, the CARs don't work anymore, but the hits still can. And in fact, if you look at CD4 and CD8 T cells looking for this time cytokine production in the presence of very low levels of CD19, you see that in red here, neither CD4 or CD8 CARs produce those cytokines, but the hit CAR T cells do. It's not unique to CD19. Here's another hit receptor specific for CD22. And this is the MM1S, multiple myeloma cell line, which actually does express trace amounts of CD19 and CD22. It's virtually negative by FACS, but the molecule is confirmed by quantitative mass spec. We know it's there. And the CAR is unable, the CD19 CAR or the CD22 CAR is unable to kill this tumor, but the track hit certainly can. I'll just finish by saying one word about the mechanism before someone asks. We don't have a full mechanism. It might take time to figure that out, but we do have some insights. And we performed this assay where we co-incubated CAR T cells engineered at the track locus with NALM6 that had been cultured in heavy amino acids for sometimes co-incubate, and at different time points, and in this case here at 10 minutes, we separate them and perform quantitative phosphoprotein mass spectrometry. And from 2 experiments, this is what we find. First of all, there's a lot more phosphoproteins in the track hit T cell than in the track CAR T cell. And here are some of those that are common to these repeated experiments. And obviously, there's components of the CD3 complex, which we're happy to see as expected. Other signaling molecules, LAT and others, that are not engaged by CAR, but are engaged by track hit. And we also saw on this list ARPC1B, which our collaborator, Claire Hivros, who's here today, by the way, pointed out to us as one being important in the mobilization of lysosomes to the synapse. So this made us think that perhaps the hit is better at degranulating and faster at killing, and I'll show you just one example of that, looking at simply degranulation. So these are track CAR T cells, track hit T cells, I'm sorry, these are track CAR T cells. We'll just look at 1 hour if you don't mind. And on the wild type NALM6 with 20,000 molecules or so, you can see that both cell types have started to degranulate rather fast. But when you go to the low line, the 20 or 30 molecules per cell, you see the hit T cell degranulating when clearly the CAR cannot. It still cannot, by the way, 3 hours later. So we think that the speed of assembly of a synapse and recruitment of lytic granules has something to do with this increased sensitivity. I'd like to discuss briefly also some efforts to increase the persistence of T cells. And here I'll go back to the 28Z CAR. This, for us, for me anyway, is a bit of an irony, having spent a decade to figure out that at last we had to put this together to produce a T cell that could proliferate, expand, and ultimately eliminate a tumor in a mouse, but the clinical data suggested to us that maybe these signals maybe are too strong, in fact. And as I think you all know, these CD28 CAR T cells don't have very long persistence. In fact, Jérôme Gallon mentioned yesterday that in his biopsies, he had trouble detecting them, sometimes as early as day 8. They do persist usually longer than that, certainly in the marrow, but their persistence in vivo is limited. And we wondered if one component of that is excessive signaling. So we started to focus not on the CD28 costimulatory part, where there's still a lot to learn, but on the ITAMs in the CD3 zeta chain, and mutated either the first or the second. So one XX retains the first, and the ITAM, the more distal ITAMs are inactive. XX3 is the reverse. And here's a sample. This has been published, so I'll be very brief. We infuse these T cells in mice bearing the NALM6 leukemia at a low dose where the classic CAR works. There's a response, but it's not very good in most mice. Obviously, this CAR works. My colleagues, Isabelle Rivière and Rainier Brantians and Jay Park have treated close to 300 patients with this CAR, but in this model, at this level, it's intentionally not very active. And you can see that these CARs that each contain only one functional ITAM differ quite a bit. The third ITAM is a poor activator, and the first one alone turns out to be the winner in this and many other models. In one experiment that I want to highlight, we took human peripheral blood T cells, naive T cells, and introduced these different CARs at the track locus. So all T cells express the CAR from the same copy number, 1, from the same promoter enhancer, expose them to antigen, and then extract RNA. And we saw that the 28Z CAR gives this very typical effector profile. In red here are a whole bunch of genes associated with effector functions. And anything that has to do with memory is essentially turned off. In contrast, the XX3, which I just showed you, was not a very good CAR in vivo. I didn't tell you that those cells happily persist in the mouse. They're just not eliminating the tumor. And indeed, they're activating, their effector functions are poorly turned on. And the 1XX kind of seems to reach some kind of a midpoint, perhaps a compromise. And today we have 2 trials that we have started with this, and it's too late really to comment on those, but I'll say that in the first 6 patients with the DLBCL that we treated, there's relapsed refractory patients again. 5 out of 6 went into complete remission, which is really to be expected for CD19 CAR therapy. The key difference is, will be relapses, but we have to wait for that. But the interesting detail is that we obtained these responses with only 25 million CAR T cells per patient, which is substantially lower than the doses used for current commercially available CARs. This told us that maybe we could think about other ways to reach this, to engineer this equilibrium between effector function and persistence. And here, this is just a teaser for 2 studies, but the principle is the following. Since there are epigenetic states that are associated with differentiation or memory or exhaustion, could we control these epigenetic states in paired fashion with the CAR, such that CAR signals would deliver the signals that we think are appropriate, but through an epigenetic programming, could we ensure that target genes remain accessible? And we're in the middle of a large, unbiased screen. But we did start with 2 candidates, one called TET2 that is very familiar to hematologists for its association with a number of actually myeloid malignancies, and another one called SUV39H1. And I'll just show you here. These are ongoing studies, not yet completed. I'll just show you perhaps a small teaser as to why we're very interested in this approach. So we knock out TET2 in these human peripheral blood T cells and then introduce a variety of CARs, actually different CARs, and treat the same NALM6 leukemia. And as you see here at these doses, low doses again, designed to not be too successful in the CAR T cell to obviate any difference, we can see that the knockout of TET2 is beneficial to the activity of these T cells. We also note that over time, there's a large accumulation of these T cells with not every CAR, and I won't go into those details, but as you see here, there's 10 to 100 times more CAR T cells 2-3 months after T cell infusion. And while this appears to be good news in the beginning because there's an improved therapeutic response, in fact, this ends up being a tremendous infiltration of CAR T cells in many tissues. And we're currently evaluating the significance of it. The other approach, targeting this methyltransferase H3K9 that methylates H3K9, is much more promising. And here we owe this study to the work of Sebastián Amigorena, who's actually also in the room today, who had shown in a very interesting paper in mouse models in ova-specific TCR, T-cell receptor transgenic mice, that the disruption of this gene could increase memory formation and blocks the transition and block the transition to effector. So we've now gone to evaluate this in human T cells, and I'll just show you. This is normal or scrambled guide RNA, and this is SUV39 RNA day 0. There's the same level. This is global methylation of H3K9. And after a few days, this methylation increases in T cells. This is on day 5, corroborating what Sebastián had found in mice. But in the knockout, it's decreased. And what you see here is, again, a very interesting increase in the potency of these T cells, and we're currently evaluating that. I'll rush through 3 more slides, I don't know. Oh, yes, it's 31. Well, I'll rush. Sorry, it's not going to be pretty. We're looking for CAR T cell sources other than the patient's own peripheral blood. The commercial products today certainly start from bulk peripheral blood cells obtained from patients by apheresis. Some groups go to T cell subsets. There's a lot of interest in developing allogeneic T cells harvested from a healthy donor. And to avoid the graft versus host reaction, different strategies are being pursued using virus-specific T cells, gamma delta T cells, invariant and KT cells, or knocking out the T cell receptors I just showed you. But a few of us are interested in producing the T cells from scratch, or at least from precursors in vitro in the absence of a thymus. And nearly 10 years ago, we had provided a first proof of principle of that when we reprogrammed T cells, expressed the CAR on a ubiquitous promoter, rederived T cells, which were T cells, but with innate features and not really the mirror of the alpha-beta T cell receptor that had been originally reprogrammed. So anyway, yes, and I'll stop. I'll just say, because we've been quietly working this for many years, not many papers on the road, but I'm pleased to say that there's an increasing understanding of the differentiation of these cells. You see them acquiring CD7, CD5, the pre-TCR comes on, RAG1 comes on, they become CD4, CD8 positive. And I think we're getting closer to that goal, and we're able to produce now T cells born in a bioreactor without requiring a human donor that are certainly lytic in vitro. And although they are not as good as human peripheral blood alpha-beta T cells, they are lytic in mouse models of leukemia. So there's still a ways to go, and we're learning a lot about Notch and pre-TCR signals and others to develop these T cells, but there is hope on the horizon. I'll stop here by thanking you for your attention, mentioning some of my long-term collaborators, and also Sebastián Amigorena and Claire Ivros, who I've already mentioned today, who are here with us today. Thank you.